Yes, next we'll see about the assay of vitamin. So here I am taking the assay of vitamin B2. As you know that it is also known as riboflavin, so which is plays a major role in carbohydrate and protein metabolism. You can get from liver, beef, yeast, peas, peanuts, etc. And if vitamin B2 is not present in sufficient amounts, you can get blood vision or dermatitis or anemia. And coming to the preparation of these standard solutions, you have to prepare some solutions, riboflavin solutions. So for stock solution, you have to prepare 50. We have to take 50 mg of riboflavin. Along with that, you have to add 350 ml of distilled water with 0.6 ml of glacial acetic acid, and we have to warm at 80 degrees centigrade. Then riboflavin is completely dissolved in that. You have to cool it and transfer into 500 ml of volumetric flask and keep it up to the mark by adding some water into that. And we have to keep that solution as stock solution A and keep it in the refrigerator. Next, this preparation contains 100 mg riboflavin per ml, which we can keep that one for about 1 month. And coming to the preparation of the stock solution pre, 10 ml of stock solution A. So first what we have prepared stock solution A. From stock solution you have to take 10 ml of stock solution A and dilute with 100 ml of distilled water, and we have to store in the refrigerator. So this preparation contains 10 microgram of riboflavin per ml. That means we are decreasing the concentration. And standard solution, coming to the standard solution, 1 ml of stock solution B we have to take and dilute to 100 ml with distilled water. This preparation contains 0.1 microgram riboflavin per ml. So this cannot be stored as the prepared on the day only, whichever we are using. Coming to the inoculum preparation, so into 10 ml of centrifugation tube, add 5 ml of basal medium and 5 ml of riboflavin solution and sterilize that one, and add lactobacillus casei, which is used as a test microorganism, and incubate for 37 degrees centigrade. Next, centrifuge it and decant of the supernatant liquid and suspend the bacterial cells in 10 ml of sterile saline. And centrifuge again and suspend the bacterial cells in 10 ml sterile saline, and then the inoculum will be getting ready. Coming to the procedure, so fill 7 test tubes with 0.5, 1.5 like that up to 3 ml of standard riboflavin solution, and make up the volume in each volume to 5 ml using distilled water, and then add 5 ml of riboflavin in each of these test tubes and sterilize the apparatus, and take another 5 test tubes and fill them 0.51 to 2.5 ml of the test samples, and make up the volume in each test tube to 5 ml using distilled water. Add 5 ml of riboflavin in each test tubes and sterilize the test tubes and cool all the test tubes to room temperature, and inoculate each test tube with a drop of inoculum, that means 0.6 ml you have to add, and incubate at 37 degrees centigrade, and you have to titrate the contents with 0.1 normal NaOH at pH 6.8. So this is your assay of vitamins. So that is only vitamin B2 vitamin I have taken, and I have performed this assay. So by this method, what you can get that, so vitamins are essential for the growth of any microorganisms. So by this method, if you are going to prepare any culture mediums, etc., if you want to grow any new strain of bacteria, compulsory vitamins plays a major role. For that purpose, we are going to do this assay of vitamins. Next, coming to the assay of amino acids. So there are 20 different types of amino acids at present, and various amino acids like alanine, arginine, methionine. So all these have specific types of species, and for all amino acids, leuconostoic mesenteroids was commonly used. This included aspartic acids like histidine, phenylalanine, proline, serine. So microorganisms are grown on the specific media and a freshly prepared amino acid solution on the same day, which is containing 200 microgram per ml with an appropriately diluted with the tubes having 0 to 1.0 ml per tube, and these two tubes are prepared using 1 ml of original levels by 0.2468 1 ml of amino acids like that. That means you have to prepare the serial dilution methods and add the amino acids in the media, and you have to perform the autoclave. That means you have to undergo the autoclave. When the media becomes cool, inoculate the test microorganisms, and you have to incubate for a certain period. That means up to 15 to 20 hours we have to incubate, and after incubation you have to compare all the test tubes with blanks having no microorganisms, and finally determine the potency of the amino acids. So without the amino acids, the bacteria cannot grow, so that plays a major role in the growth of any bacteria. So this is about your microbiological assay of antibiotics.